R is a statistical programming language used to plan, analyze, and report clinical trials. It matters because one environment can handle data preparation, modeling, and graphics, then produce inspection-ready tables, listings, and figures. This reduces handoffs, speeds iteration, and keeps work traceable. For programmers, R offers flexible tools and strong integration with version control. For the general public, the benefit is simpler. It helps researchers turn trial data into reliable, transparent evidence that regulators and clinicians can trust. You use R throughout the study lifecycle. In planning, it runs simulations and power calculations so teams can test sample size and event rate assumptions before the first participant enrolls. During conduct, it scripts data checks, merges, and derivations, so the same logic runs each time new data arrives. For analysis, it handles summaries and models, from time-to-event methods such as Kaplan-Meier curves and Cox proportional hazards, to generalized linear models for binary outcomes and mixed effects models for repeated measures. For safety, it produces adverse event summaries and exposure-adjusted incidence rates with clear stratification. In reporting, it creates tables and figures and exports them to Word or PDF using reproducible code, keeping outputs consistent. Here is how the core workflow fits together. Teams start from standardized analysis datasets, often aligned with CDISC ADaM, so variable names and derivations are predictable. They create a reporting dataset that mirrors the final table. It includes row order, labels, indentation levels, group identifiers, and any planned blank lines. A few functions convert that dataset into a formatted table and write it to a Word document with approved styles. Figures follow the same principle. Build tidy datasets of estimates and confidence intervals, apply a reusable theme for fonts and spacing, and export at fixed dimensions. Version control tracks every change. Session information records the exact R and package versions used, so results can be reproduced later. This approach satisfies regulatory expectations for data integrity, traceability, and validation with clear documentation and testing. To prepare for implementation, define an internal procedure for how R is used. Pin the versions of R and its packages for each study and store them in a controlled environment. Agree a clear project structure with folders for raw data, processed data, scripts, outputs, and logs. Establish naming conventions and a style guide for code, tables, and figures. Train the team in tidy data manipulation, model fitting, and the reporting toolchain. Start with a small pilot, one demographics table, one adverse event summary, and one time-to-event figure, and compare outputs with your current process. Resolve any differences in defaults or rounding and document the decisions. Build one reusable function to create house-style tables and one theme for figures, then apply them across shells to enforce consistency. Keep a quick checklist in mind. Set up standard operating procedures that describe purpose, inputs, outputs, and acceptance criteria for each script. Record session information with every deliverable to recreate the exact runtime. Validate reporting templates so headers, footers, fonts, and pagination behave as specified. Apply independent code review or dual programming where risk is higher and keep the evidence. Use a reference Word document to control styles and write tables into it programmatically. Control access with role-based permissions and an audit trail in your repository. Align your analysis datasets with agreed standards and store the specifications with the code. For figures, use a reusable theme and fixed export sizes for consistent appearance. Be aware of common pitfalls. Leaving layout to the final printing step slows teams down. Fix structure in the dataset first, and formatting becomes simple. Mixing package versions within a project undermines reproducibility. Lock versions from the start, and avoid mid-study upgrades. Inconsistent naming and file organization create confusion. Adopt a standard template for projects and stick to it. Skipping independent review of simple scripts invites small but important errors. Schedule a light, consistent review. Upgrading packages without a plan can change defaults and shift results. If you must update, run a controlled comparison on defined outputs, document differences, and seek approval before deploying. Plan safety summaries early, including exposure-adjusted rates and stratifications. Open source does not remove validation duties. Validate process and environment as you would with commercial tools. If you are weighing R against established clinical software, focus on process rather than brand. Both can produce compliant outputs. R is strong in flexible modeling, graphics, automation, and integration with version control and reproducible documents. Manage trade-offs with training, clear procedures, and pinned environments. Main costs are upskilling, process design, and maintenance, not licenses. With a careful pilot and disciplined workflow, R becomes a reliable part of clinical reporting rather than a parallel experiment. To learn how Quantikit can support your next trial, email inquiries@quantikit.com or submit an RFI through our website.